Cancer is a complex disease. It involves more than just uncontrolled cell growth. Scientists now recognize the crucial role of the tumor microenvironment. This environment includes surrounding cells, blood vessels, and signaling molecules. Among these players, cancer-associated fibroblasts, or CAFs, have emerged as key collaborators in tumor development. CAFs are a diverse group of cells found in the stroma, which is the connective tissue surrounding tumor cells. They are not cancerous themselves, but they actively participate in tumor growth and spread. Think of them as architects and engineers of the tumor microenvironment. They provide structural support, nutrients, and signaling molecules that tumor cells need to thrive. Understanding CAFs is crucial for developing new and effective cancer therapies. CAFs are involved in various stages of cancer progression. They promote tumor cell proliferation, survival, invasion, and metastasis. They also contribute to drug resistance, making treatment more challenging. The presence of CAFs in tumors is often associated with a poor prognosis for patients. Scientists are actively investigating the complex roles of CAFs in cancer. They aim to identify new therapeutic targets. By disrupting the activity of CAFs, they hope to develop more effective treatments. The goal is to improve outcomes for cancer patients. CAFs are like master builders, constantly remodeling the tumor microenvironment to favor cancer cell growth. They secrete a variety of substances that act as signals and building materials. These substances create a supportive niche for tumor cells to thrive. One of the key functions of CAFs is to produce extracellular matrix, or ECM, components. The ECM is a complex network of proteins and other molecules that surrounds and supports cells. CAFs secrete collagen, fibronectin, and other ECM proteins. These proteins provide structural support to the tumor, allowing it to grow and invade surrounding tissues. But CAFs do more than just provide scaffolding. They also release growth factors and cytokines, which are signaling molecules that influence cell behavior. These factors, such as TGF-B, VEGF, and HGF, promote tumor cell proliferation, survival, and migration. They also stimulate angiogenesis, the formation of new blood vessels, which is essential for tumor growth and metastasis. The tumor microenvironment is a dynamic and complex ecosystem. CAFs play a central role in shaping this environment to the advantage of cancer cells. They provide both the physical framework and the molecular signals that fuel tumor progression. Understanding how CAFs orchestrate these processes is crucial for developing strategies to disrupt their activity and inhibit tumor growth. Studying CAFs in the lab is essential for understanding their biology and developing potential therapies. However, there are key differences between CAFs grown in culture and those found within the tumor microenvironment. These differences highlight the adaptable nature of CAFs and the challenges in translating lab findings to clinical applications. The lab environment does not fully replicate the complex three-dimensional architecture and dynamic signaling of the tumor microenvironment. Within the tumor, CAFs are exposed to a constantly changing environment. They interact with various cell types, including tumor cells, immune cells, and blood vessels. They also experience fluctuations in oxygen levels, nutrient availability, and mechanical forces. CAFs at the tumor interface often exhibit distinct characteristics compared to those further away in the tumor stroma. Cancer-associated fibroblasts are not a homogeneous group. They display significant diversity in their origins, characteristics, and functions within the tumor microenvironment. Researchers have identified several subsets of CAFs, each with distinct markers and roles in cancer progression. Two major subsets of CAFs are myofibroblastic CAFs, or MyCAFs, and inflammatory CAFs, or ICAFs. MyCAFs are characterized by their expression of alpha-smooth muscle actin, or ASMA, a protein typically found in smooth muscle cells. They are often found at the invasive front of tumors, where they promote tumor cell invasion and metastasis. MyCAFs are highly contractile, generating forces that remodel the ECM and create tracks for tumor cells to migrate. ICAFs, on the other hand, are characterized by their expression of inflammatory cytokines, such as IL-6 and IL-8. They are often found in regions of the tumor with high immune cell infiltration. ICAFs contribute to the inflammatory microenvironment, which can promote tumor growth, angiogenesis, and immune suppression. They also play a role in recruiting immune cells to the tumor, although these immune cells may not always be effective in controlling tumor growth. The identification and characterization of CAF subsets are crucial for understanding their specific contributions to tumor progression. This knowledge can guide the development of targeted therapies aimed at selectively eliminating or modulating the activity of specific calf populations to inhibit tumor growth and metastasis.